Hello dear students. In today's class, we'll be continuing our studies on cell interaction that comes under cell biology. Then moving on to the types of anchoring junctions, they are adherents, desmosomes and hemidesmosomes. We'll be studying in detail of all of these in the further slides. So next we'll be studying about adherence. So moving on to the study of this adherence. The adherents are the type of the cell junction that are involved during the cell-to-cell interaction or cell-to-matrix interaction. During a cell-to-cell interaction, that is intracellular interaction, the protein that is involved, intracellular protein that are involved during the cell-to-cell interaction is an actin. And transmembrane proteins are also involved in adherent junction, that is cadherin, when there is a cell to cell interaction. And when there is a cell to matrix interaction, then the protein, that is transmembrane protein, which is involved is a integrin. And they take part in different situation. Cadherin involves during cell to cell interaction and integrin involves during cell to matrix interaction. And moving on to the types of the adherents, they are adhesion belt as well as focal adhesion. So this will be studying in the next slides. Let us know what is adhesion belt. Adhesion belt is also called as zona adherens, which means that the two cells adhere with each other or stick with each other. Let us see how they stick with each other to transfer the information. So here are the diagrammatic representation of this adhesion belt. Here you can see that there are two cells which are adhered or attached with each other or sticked with each other. Because in both the cells there are certain proteins which are helping them to stick with each other, like actin filaments and cadherin dimers and anchor proteins. So in the cytoplasmic region of both the cells, you can see the actin filament, which are the intracellular proteins. The intracellular proteins, it's the actin filaments. The other filaments of this actin filament are catenins, vinculin and alpha actinin. And also the transmembrane protein that are involved in the adhesion belt is a cadherin dimers. And these cadherin dimers and the actin filaments, that is transmembrane protein and the intracellular protein. The intracellular proteins are nothing but that are present inside the cytoplasm of the cells. See, in both the cells you can see. So the intracellular protein and the transmembrane protein, which is a cadherin protein, they are linked by another protein called as anchor protein. So the actin filament and the cadherin dimers are linked by the anchor protein. So this is the extracellular space of the cell. So the extracellular domain of the transmembrane region of both the cells get fused to form a cadherin dimers. So you can see that cadherin dimers are formed. These cadherin dimers help the cell to fuse with each other. This is how the cells adhere with each other, so that the multiple cells adhere with each other, that forms a adhesion belt. Here also you can see the adhesion belt. This is the enlarged picture of this adhesion belt where the cadherins are there. Okay, there are bundle of actin filaments. See the red one here. The bundle of actin filaments are also present, and this is the upper region of the cell where actually this is present. Actin filaments inside the microvillus. Okay, this is the basal surface here. Now let's see what is focal adhesion. Focal adhesion means joining of cell to matrix. This is the pictorial representation of focal adhesion. In the focal adhesion, you can see that there are different types of proteins involved for the cell to matrix interaction. This is the cell and this is the matrix region of the cell where they are attached with each other with certain different kinds of proteins. Here you can see that the intracellular protein which is present in the cytoplasmic region of the cell is the actin filament. The actin filament along with this other types of actin filament are also involved, that is alpha actin, vinculin and talin. And here the transmembrane protein which are present is a integrins. These integrins have a two subunit, that is alpha subunit and beta subunit, but among them the beta subunit is attached to the extracellular matrix, and then the other domain of the transmembrane protein, here you can see that this is attached to the actin filament. Alpha-actinin is attached to the actin filament, and this is how the cell to matrix interaction takes place, and this is the immunofluorescence image of the focal adhesion. Okay, till now we have learnt about the adhesion belt and focal adhesion. In the coming slides we will be dealing about desmosomes as well as the hemidesmosomes.